Thank you, everybody, for your attendance today. Hopefully, everyone who's joining us today for the second session was able to join us for our first webinar based on common laboratory statistics for the performance of laboratory tests. Somebody did ask a question, are these seminars or webinars available to review if you're, want to review them again? And yes, they are. So we obviously send out an email link to everyone who wishes to see them again, and we'll have this arranged after this event. So just before we start with Stephen, thank you everyone for joining us today. This is the Applied Statistics in the Medical Laboratory, Analytical Performance Specifications. I don't think I could say that more than 3 times. So I'm glad it's Stephen taking the session today. So if anyone isn't aware of Stephen, we're delighted to have him back for his second session. Principal Clinical Scientist at Cambridge University Hospitals. Stephen is probably very well known across the UK, and we're delighted to have used Stephen's expertise over the last few years regarding IQC, EQA, and the various statistics around this. So without further ado, I will pass you over to Stephen. And sorry, just before we do, this is quite, I appreciate people might have questions, Q&A and chat. We will do our best to see if there is time at the end of the session to answer some of these questions. However, if not, there will be an opportunity for you to give us a feedback form. If you have questions that you would wish to ask Stephen, please do that in the feedback form, and we'll do our best to make sure all questions are answered. So thank you, and I'm going to pass you over to Stephen.
Good afternoon, everybody. Thank you all for joining. As Susan says, this is the second in a planned series of 3 webinars talking about some of the applications of statistics in medical laboratories and maybe looking at things slightly differently. The first session was very much based upon the building blocks for much of what we do in the lab as far as stats is concerned. So our introductory statistics on means, standard deviations, coefficients of variation, measurement uncertainty, and total error. So what we're going to talk about in the second session is how we start applying some of those statistics to determine whether our assays are any good or not. So specifically, the title is Choosing Analytical Performance Specifications. So we're going to be talking about models again, and I showed a picture of George Box at the tail end of the last webinar with his famous quote that all models are wrong, but some are useful. And I think it's very much worth reminding ourselves of that point when we start out on this discussion that we're going to have today, that there are many different ways of doing this, that there are developing aspects in the field and there are more established aspects in the field. And really, what we need to do is try and balance the input from both of those or of all of those aspects and try and work out what suits each of our individual tests as a whole. We're not going to be able to do a one-size-fits-all modeling approach for analytical performance specifications for all of our tests. So we really need to have a more scientific approach to working out how we're going to do it. So let's start off with analytical performance specifications and ask some questions about them. First of all, what are they? As I said, they're basically models, and they're even described as models in some of the approaches that's taken. And we're trying to characterize what is a good performance or what is an expected performance for a clinical assay. And they're absolutely needed for understanding whether the quality of our tests is to the desired standard to maximize positive patient outcomes. Where are they used? They are used throughout the whole of laboratory science. And not only do we have to go through the first hurdle, which this webinar is about, which is choosing analytical performance specifications, we then need to work out where we're going to apply them and which models suit different applications. And finally, what is the right one? I'm not going to be able to answer that today. That's not something that's able to be answered. I think, as I said, there are different aspects to all of the different approaches which are taken for APSs, and the long and short of that is we have to work out which ones best suit our clinical disciplines on their own. They provide us with some objective information about the quality of the lab's individual performance, and that's essential. That's something that we're very much used to doing with internal quality control and EQA. But what APS or performance specifications try to do is combine that into a single measure so that we can ultimately drive improvement in our quality if it's not quite up to the clinical requirements, or even in the furthest extreme, to withdraw assays that are not up to standard. Because if we're running tests which we know are not performing correctly for what our patients require, then we have to have some sort of gauge to measure them against. Ultimately, the question that we ask and the way that we determine performance specifications is by asking the question of how good does a test need to be to do more good than harm to a given patient? And that's a very high level look at it. But ultimately, we want to be ensuring that the information that we give to our clinicians is of value to those clinicians and can aid them throughout the pathway of a patient treatment. Because remember, we are providing data to clinicians. Those clinicians are then instigating clinical interventions or withholding clinical interventions based on those results. So it's a very indirect link that the performance of assays have to the final patient outcome. And that becomes really challenging when we're trying to set performance specifications, because trying to find the evidence that the performance of our assay actually improves the diagnostic yield. What do I mean by that? Increasing the true positives, reducing false positives. It's a very indirect link, because ultimately the information that we're giving is going to be adding to further things down the line that are being enacted. We also have to determine, as I said, the therapeutic yield, the therapeutic intervention, so that we can support appropriate treatment and reduce inappropriate treatment. So what do they look like? In the simplest terms, performance specifications are criteria that specify, using numerical terms, using a number, what quality of our assays is required and the quality of analytical performance, so that we can provide results that support clinical need and improve health outcomes. And central to all of that is this slide that I showed in the very first webinar, where loosely called the Temple of Standardization, but where we talk about the various different pillars which are intertwined and linked together to be able to allow us to provide standardized assays so that we can then provide performance specifications based on that. The interesting point is that this standardization process both requires and provides the utility of performance specifications. And without standardization, we can't actually provide performance specifications. By then providing performance specifications, of course, we can then drive things further towards harmonization and ultimately standardization. The last column here, which is talking about measurement uncertainty and error of measurement, which is effectively what we're talking about when we talk about performance specifications, they feed back to the other pillars as well, because by setting the limits based on our modeling approaches, we can then work out what processes we need to input or what we need to instigate to be able to monitor conformance to those performance specifications. So I think the point is that it is a very fluid process. It's a very interactive process between all of these pillars. It's certainly not sequential. And when we have something that then feeds back into processes previously, that's able to then drive the standardization forward. So if we talk about performance specifications, ultimately, and we have to be careful of the terminology that we use here, if we talk about measurement error, and we'll use that as an introductory term and we'll diverge on that later. Measurement error, as I'm sure everybody is aware, can basically be subdivided into contributions from two different sources. On the diagram here, which is adopted from the CLSI EP 21A guideline, if you imagine our ultimate goal is to try and measure this true value over here on the right-hand side in the purple dot. By trying to implement a measurement process, we're then trying to determine what the value of that true value is. but we accept there's always going to be some source of error within there, and there's going to be multiple sources of error within there. And we classify them as either random error or systematic error. So systematic error is also known as bias. We use this line, this arrow here, which basically quantifies the difference between our measured value and the true value. So that's an absolute value, that's an absolute amount away from where the true value lies that we're actually measure and quantify that result as being. However, we do accept that what we do as a measurement is only a point estimate of what the potential values of that measurement could be. And actually, the result that we're providing is the most likely expectation of the result that we have. So I mentioned that in webinar 1 again, the expectation in a normal distribution is synonymous with the mean value. But we know if we run this measurement multiple times, we are likely to get a normal distribution of results around what we identify as the mean value. And the degree of spread measured by the standard deviation is an indication of the random error which is associated in that measurement. Now, of course, these don't work completely independently. So every measurement will have an amount of bias. Every measurement will have an amount of random error in there. And it's the combination and the magnitude of those two things together which determine the quality of the result against the true value. So we can see here that actually, if the true value is away over on the right-hand side, there's a reasonable probability that way over where this black line is on the left-hand side, that a result could lie. And that magnitude can be quantified by the extent of the bias here, plus a description of the normal distribution, 1.96 times the standard deviation. That's a two-sided measure of the standard deviation, and we'll talk about that, the difference between a two-sided and a one-sided measure. But the point is that basically we combine these different sources of error together to give us some sort of measure of the analytical quality. And for many years, up until in fact the mid-70s, imprecision and bias were very much considered as separate entities, and they were determined in the laboratory separately. And it wasn't until the mid-70s when this paper by Dr. Westgard and colleagues showed that there was a way that they could actually combine the random analytic error and the systematic analytic error to give a term which is called the total analytic error. And this total analytic error is probably a term that is familiar to many people on the call just now, has been the mainstay of describing the quality of results for many, many years. But time passes, and as time goes on, lots and lots of other terminologies have been introduced into the language that we have. Not quite to the extent of the number of different words for snow that the Inuits have, but you can see on the left-hand side here, there is a long list of what are effectively analytical performance specifications, APSs, which are termed in various different ways similarly. So total error, total analytic error. Based on total analytic error, we can define a target that we set, which is called total allowable error, or is also known as total error allowable or allowable total error. It gets very confusing. There's lots and lots of different terms used. But the total error approach was very much the mainstay from the mid-70s up until round about the late 90s when measurement uncertainty came onto the landscape, and that gave a different approach and a different way of looking at performance specifications, which, along with total error, are now the ways that we understand how to apply it in our systems. Measurement uncertainty, of course, is not just one single term. There's also uncertainty of measurement. It is not measurement of uncertainty, which is a phrase that I hear an awful lot, but that's not a phrase that is actually associated with performance specifications, although it seems to be a common phrase that's used. And if we're looking for performance specifications, there are many associated with measurement uncertainty, but maximum allowable uncertainty is one, Umax is another, and we'll discuss them later on. But as I said, total error, following the work of Dr. Westgard, was very much the mainstay of determining analytical quality in the laboratory. And if we use total analytical error as the acronym that we'll use of choice, the way that that is calculated is by determining the absolute value of the bias. So those vertical lines, each side of the bias means it's the absolute value, which means whether it's positive bias or negative bias, the sign is disregarded. It's just the actual size of the bias that's included. We then add that to a statistical factor, which describes the probability distribution of the random error. So that may be 1.96 or 1.65. And then the measure of the standard deviation of the laboratory's performance within the laboratory, called the standard deviation within the laboratory. And from there, we can then determine what the analytical error within the measurement processes in the laboratory are quantifiably, and we can compare that against a limit, which would be the total allowable error or the total error allowable. As I say, that this has been the mainstay for many, many years, and has been the basis upon which quality specifications have been based. I have to do a talk and loop back to webinar 1 and talk about cookie specifications. To compare that to what we do if we're baking cookies, for example, we have lots of different specifications which we're trying to achieve, different quality markers which we're measuring, texture, flavor, appearance, etc., etc. But ultimately, the outcome is the same thing, irrespective of what we're describing in the list above. Qualitatively, we're talking about the quality of the cookies. So how do we determine that in laboratory testing? Again, looping back to the first webinar, how well can we diagnose a given condition? And the mainstay of diagnosing a given condition is, of course, clinical decision limits or reference ranges. And how our assays perform will ultimately determine what the probability of a patient being correctly diagnosed is. In addition to that, we can also determine how well we can detect a change in the clinical situation. So I showed this graph in the first webinar, where if we determine a reference range from, let's say, the central 95% of what would be a reference population, we know that normal patients may well creep outside of that normal range, but once we start implement or adding in a bias, we get an exponential increase in the amount of absolutely normal patients which are then being classified as abnormal. So this isn't a linear response, this is an exponential response. And I quoted some numbers last time that a bias of 0.5 SDs will give a further 10% being moved out of that reference range. So it's really important that we control bias and we understand that and measure it, because ultimately it's going to have a really significant impact on misdiagnosis. And equally so will changing the standard deviation or the random variation in our tests, comparing it to a reference limit. You can see the tails of these distributions get significantly heavier. If the standard deviation increases, the expectation of what the result will be lower, and the probability of it being outside that range, perhaps erroneously, due to the analytical variation, will be increased. So over the years, there's been a lot of discussions about how can we actually link performance specifications in our assays to the actual clinical outcome of patients and the clinical behaviour of the measurands that we're measuring. And this conversation started way back in the early 1960s and has progressed through a number of different conferences and consensus agreements, ultimately culminated in 2014 in the first EFLM Strategic Conference consensus statement, which came from a meeting in Milan in late 2014 and was laterly published in 2015. This built on a previous meeting from the IFCC in 1999 held in Stockholm, and it narrowed down the models that were used for performance specifications from the 5 in Stockholm down to 3, which were then taken from the Milan conference. These are Model 1, Model 2, and Model 3, each of which we'll walk through individually. Model 1 is actually subdivided, but that takes a look at towards how does the performance of our tests directly impact clinical outcome or indirectly impact clinical outcome by influencing medical decisions. Model 2, which is probably by far and away the most familiar to most people, is based around biological variation and has been the mainstay of the total error approach for a long, long time. And they've also added in a third model, which is state of the art. So that is the best possible analytical performance based on what was available on the market at the time. And Cheriotti in 2017, following that meeting, developed this algorithm which allowed us to work out how to apply different measurements to different models. So rather than thinking it as a hierarchy as it is shown here, models 1 and 2 are actually considered to be equal, and determination of which model you use has to be based on the clinical utility of that measurement. So the first question, has the measurement a central role in a specific disease? So diagnostic of a specific disease and will impact clinical intervention in those diseases. We then check for valid outcome data and whether that exists, and then we can assign it to the outcome model. That valid outcome data must be related to the clinical outcome being linked to the analytical performance of assays, and that is actually vanishingly rare, and we'll discuss that shortly. We can also look at the biological variation and determining whether the measurand we're measuring has a steady state in the human body and whether we have valid biological variation data existing, in which case we can apply to the model. So those two models are held with equal weighting. Depending on the clinical use of the assay, we choose one or the other, and depending on the data available, we choose one or the other. If none of that applies, we then have to go to the state-of-the-art model, which is basically the best that we can do in our laboratories routinely. So model 1 can be described as the clinical outcome model, and that can either be from direct outcome studies, again, very difficult to evidence, or more commonly from model 1B, which is through indirect or simulation of if our performance gets worse, what is the potential outcome to the patient going to be if it does get to a certain level. And there's been a lot of activity in this field, even in the last couple of days. I've got a Google Scholar result with some more publications about this model. As I said, Model 1A, directly linking performance to clinical outcomes, has been described as being aspirational because the data is not really there. And in order to generate that data, we might need to think about running randomized control trials based on determining clinical outcomes of patients used on different assays with different performance specifications. That's not something that's going to be particularly simple to do. So usually the Model B, the simulation approach, links more of if the analytical performance degrades over time, how does that impact the interpretation by the clinicians? And in doing so, does that influence their decision about further treatments and interventions further down the line? So it's not directly linking the performance specification to the outcome as Model 1A is, it's more through this indirect linkage that we see here. But we do have measurements that are linked to Model 1. And again, these are through simulation data. And after the conference in 2014 and the publication in 2015, the work began with trying to assign different measurements to different models. So a good example for the Model 1 measurements is the high sensitivity cardiac troponin, where it was simulated that a coefficient of variation less than 10% will result in a misclassification error for patients with acute coronary syndrome of 1%, and that's considered acceptable. So that's an important point to raise, is that there has to be clinical input into what the acceptable risk of misclassification is, and therefore linking that to the clinical outcome. Misclassification can be further improved to 0.5% if the assays were available to come down to a CV of less than 6%. And also, with the other extreme of the spectrum, the misclassification rate can be vastly inflated again, non-linearly, based if the CV goes up to even as far as 16%. So we can see in these examples here that having the joined up thinking of what is the clinical utility of the test, How was it used? Was the acceptable risk of misclassification? Ideally, that would be 0, but realistically, what sort of level do we need to sit our analytical performance specifications at so that we can hit that target? Interestingly, and we'll talk about CRP later on, interestingly, CRP was initially touted as potentially being a Model 1 measure, and that's been changed now, but that was stressed within the 2017 paper that it's only for specific clinical uses only. So CRP is, as I'm sure most people are aware, one of the most commonly ordered tests in medical laboratories, used in many, many different applications. So the use of it using Model 1 was specified for specific clinical uses only, but that's been superseded since. Not least by this paper that was published by Braga and Pantaghini, where they, in 2021, took some of the most commonly selected biochemistry measurands and assigned them to different models based on the criteria from the Milan meeting, and determined what the APS, the analytical performance specification for standard measurement uncertainty, should be based on the models and the data available. So, for example, we see up here, if we're talking about the outcome-based model, so Model 1, plasma glucose has a desirable measurement uncertainty of 2%, minimum of 3, and that is based on studies at 276 mg per deciliter, largely coming from studies by Heilhoff-Peterson, where he used simulation misclassification of patients that were known to be diabetic and they were being misclassified as normal. When they had unbiased results, a coefficient of variation of 2% gave the false negative and false positive rates of 5%, which were considered acceptable. So that's how this 2% desirable criteria was derived. Hemoglobin A1C is another example, which has been very well characterized when talking about performance specifications, in that, again, used for diabetes and diabetes monitoring. They found that for the 3% of patients, of diabetes patients, were undiagnosed when the CV was 2%. This increased to 3.7% when the CV increased itself to 3.7%. So they've set the CV analytical performance specification to 3 there as the desirable. Some recent publications from the group of Low et al. Have done, so again, some simulation studies, and it's showing the same sort of thing. The graphs on the left-hand side are from this publication, which has only very, very recently come out, showing the variation in the performance of the assays and the misclassification rate of patients based on protein assays. What we really need to look at in these graphs is there's not a linear response. And this follows on from work that they published previously in this paper, that reclassifications, or more correctly, misclassifications, increase with the magnitude of imprecision and bias in our assays. That makes a lot of sense. That completely makes sense. But the important thing is that imprecision is less impactful when bias increases. So this linear combination of imprecision and bias now starts to look like we need to dig into it a little bit further. We can see that quite nicely in the recent publication, where the different colored lines in the top graph represent the misclassification rates as we increase the random error. So lower random error obviously has the lowest misclassification rate, even in the presence of a low bias at the lower limit of classification, so the lower limit of the reference range. What's interesting is that there's the convergence of the misclassification rate and the divergence based on what the magnitude of the bias is. Now, by setting an acceptable misclassification rate of 10%, they've said that obviously we can now determine what CV and what bias we can accept within our assays to minimize that misclassification rate below the level that we're looking for. And that's really, really important work, because it now gets us away from thinking about linear combinations of different sources of error. That what the magnitude of the imprecision is exquisitely related to what the bias is. And by combining maximum allowable error from both imprecision and from bias needs to be thought of very, very carefully. And in doing so, it makes us think more about the use of our assays, because using different assays and having acceptable misclassification rates means that we can have a bit more freedom in what we're trying to do with selecting our assays. Particularly with the clinical utility of the assays. So in this paper by Schmidt et al., they compared two different assays used to quantify creatinine, and they noted that there was a difference in the performance, and the bias was being introduced by interfering substances, which severely impacted the diagnostic accuracy measure, specificity in this case, and that importantly, that effect is greatest at the decision limit. And therefore, because it's at the decision limit, it has the highest risk for patient outcome. Now, what we need to think about is actually how is that test used? And their conclusion was that the context that they were measuring it in chronic kidney disease, the diagnosis is not based on EGFR alone, of which creatinine is a component. So there are other things which contribute to that diagnostic criteria. So the individual assay performance and the risk of misclassification has to be thought of in the context of that clinical utility. And actually, they found that biological variation has a more significant impact on the misclassification rate. And that leads us nicely onto the second model. So biological variation, again, probably something that is more familiar with many people for developing performance specifications, generally can be thought of as being derived from two different components. If you imagine every measurand within an individual will have what is called a homeostatic set point. So if I measure an individual measurand within me, it will usually run at a given level in health in the steady state. Now, of course, that varies around that set point to a certain extent, but shouldn't be to too much, depending on the measurand, of course. So that biological variation within me is the intra-individual biological variation, the CVI. Now, my homeostatic set point will be completely different to an extent to everybody else that's on this call just now. So everybody else will have their own homeostatic set point, and they will all be different. So the degree to which they vary across the entire population of the people on this call is the inter-individual biological variation, CVG. So it's CVI for individual, CVG for group. And these different parameters can be determined using prospective studies by sampling normal donors. And they can be measured as single measures, or they can be combined to give us combined parameters which can give us targets for our performance. Biological variation as a performance specification is basically used to ensure that our analytical systems are to such an extent are working to such a quality that the analytical variation is significantly less than the biological variation. So the signal to noise ratio is significantly different, so that any change in the biological behavior of that measurand within an individual can be easily detected and isn't masked by analytical variation being excessive. It does, however, not have a link to clinical use of the test, because the data which is used for this is basically used in steady state patients in health. So we need to be careful how we use the data in unhealthy subjects. We apply biological variation for our analytical performance specifications if they don't have a central role in a specific disease. If they have a central role in the diagnosis of a specific disease, we go back to the Model 1 models. So how does that look in our model assignment workflow? So has the measurand a central role in a specific disease? We say no, and then that drives us through checking for valid biological variation data. And if there is valid biological variation data, then considering using that for performance specification derivation. And I've stressed that point about valid biological variation data because there has been a long-standing history of data being available, but how we interpret that data and how we use that data has changed significantly in recent years. not least due to the harmonisation of the biological variation information held on the EFLM biological variation database. What they have done is they haven't taken all of the studies on biological variation and calculated the parameters from all of that. They have gone through every single study which has been published on every single measurand for biological variation and scored it based on a template to determine the quality of the data. And in doing so, we can now see from the data that is actually produced how applicable that is to the analytical performance of our tests. It's a massive, massive change in the way that we think. The variation database is available from this link here. Literally plug it in, up in the search bar, up here, plug in any measurand, and that will give you not only the data for the individual studies about biological variation, but a harmonized and combined number, which will tell you what the expected biological variation on good quality data will be for each measurand. An example of which is this. So I think this was sodium that I chose in the end. There is another tab where you can click for the analytical performance specifications, and it summarizes it all in a nice tidy number, both using the maximum allowable measurement uncertainty and also the total error approach. So this is really a big step change and is really going to help how performance specifications are viewed moving forward. Going back to the study from 2021, excuse me, you'll see of the most commonly requested measurands that were tested in this paper, most of them fell into the biological variation category. So we've got some electrolytes in there and some kidney function tests and things like that in there as well. But by using the biological variation data, just the same as we did in the outcome-based, we can have desirable criteria and minimum criteria calculated in different ways, which we'll talk about in the next slide. So that gives us the target for what we should be trying to achieve in the laboratory. So to look at sodium in a bit more detail, we can see that the intra-individual biological variation is 0.5%, and between the group, the inter-individual biological variation is 1%, giving us these targets of 0.8%, 0.5, or 0.3 based on whether we're trying to go for minimal, desirable, or optimal. So where do they actually come from? If we're trying to work out what our ideal in precision, so our analytical coefficient of variation is, that's calculated as taking the intra biological variation and multiplying it by a half. And that gives us our desirable criteria. Now, obviously, not a half times 0.5, there's some rounding going on there, but the target of a 0.3% CV for the imprecision of a sodium assay is the desirable target that we should be aiming for according to this model. Bias combines both intra and inter biological variation through the root sum of squares method that we mentioned in the first webinar, and multiplies that by 0.25 to give an allowable bias for desirable criteria again. So if you wanted to do the maths in your head, you would again get a result of acceptable bias of 0.3%. From that imprecision and bias, we can then calculate what the maximum allowable uncertainty is. So the maximum allowable uncertainty is a coverage factor of 2 to allow us a 95% coverage interval, multiplied by the CVI, multiplied by 0.5. So you can see that result here. And the total allowable error is from 1.65, because it's a one-sided distribution that's used, multiplied by the imprecision allowance plus the bias allowance, and that is added linearly. But there are challenges. Because some measurands are under very tight homeostatic control, such as sodium, which we all understand, the limits based on this can be very, very tight and very, very difficult to achieve for IVD manufacturers, but also for us in the laboratory. And a really important point is also that the biological variation studies need to be fit for purpose. And reference 2 down here, Crintis and Pantaghini, which is an absolutely tremendous paper in my opinion, really highlighted how biological variation studies have to be designed with the clinical use of the assay in mind first. There's no point in measuring what the weekly variation of a troponin result is if you're trying to do hourly measurements in an acute coronary syndrome patient. And he found that, or they found that, the biological variation for troponin, a lot of it was actually irrelevant because it wasn't using the clinical use of the test. So the biological variation model is still used for the lion's share of a lot of tests that we use. But there are challenges with plasma and sodium, as I said. Even looking at the minimum criteria, we are, or certainly the laboratory that produced this publication, are failing to achieve those minimum specifications. And there are reasons for that, which we'll talk about how to fix later on. So the final model is the state of the art. So that is the best possible analytical performance that we're able to produce. And the example you'll see here, the reclassification of the C-reactive protein into this model now, because we're now understood that CRP, as we know, is released in response to cytokines, part of the inflammatory response, was in fact the first acute phase protein to be described. So it's not got a central role in a specific disease, although it does have some specific clinical uses that it's in. So we then move on to, is there any biological variation data that exists? And we can look at from the database here what the values are for the intra and inter biological variation in the CRP. That is massive. So a 34% intra individual biological variation and 83.6 for the inter, so across individuals. This is for a simple CRP assay. For a high sensitivity CRP, it gets even worse. And what you can see is from the performance specifications we're trying to hit, saying that we can have an allowable total error of 100% in our results is completely unsatisfactory. So that would suggest that the biological variation model is inappropriate because the data isn't really fit for purpose, and it shows the biological variation can't be applied to all measurements. Why might that be? If we get an intra-biological variation of greater than about 33%, we think it might be due to a log normally distributed measurand at rest. But also for the CRP studies in particular, it was thought that a lot of the people that are being tested for the biological variation studies may have a subclinical transitory inflammatory response going on, whereas they might feel normal and everything, they might have a spike in their CRP, which grossly skews the data. So for that reason, we're taking out the outcome model, we've taken out the biological variation model, and we go to the state of the art model. So the best that we can possibly achieve. So how do we go about doing that? How do we know what the best state of the art is within the discipline that we're working in? So first of all, we've got to identify what measurement systems are in use. So the most commonly used analyzer and reagent combinations. We can calculate the imprecision within the laboratory, the long-term imprecision or the intermediate imprecision in the form of URW from our IQC performance. Work out what the calibration uncertainty is, so we're making sure that the traceability hierarchy is included within our considerations here. And we can combine that based on the equation that I showed in the very first webinar into an uncertainty of the final result. And from that, and by comparison of the different measurement technologies that we have, we can identify what the best one is based on current performance. And we take that as our desirable performance specification. We can then multiply that by 1.5, so adding a 50% uplift, and we should be setting that as the minimum performance specification. So again, the group of Braga and Pantaghini published this in 2020 for CRP assays. And you can see here that, first of all, thankfully, they took measuring the standard uncertainty for random sources at a clinically important level. So detecting subclinical infection at the level of 10 mg per liter. So we can apply this to the clinical use of the test. But we can see across these four different methods, there is a difference in what the within laboratory performance is. So this is just how the performance of the quality control is performing across the different methods, nearly a factor of twofold difference across. In addition, the combined uncertainty of the calibrator value is slightly different, and this is despite them all using the same traceable reference plasma for each of the different methods. So how are they different then? How is the question? So if we have a reference standard or certified reference material with an uncertainty assigned to it, our IVD manufacturers will then use their manufacturer calibrator and use that as a traceability against that reference plasma. The method that they use to assign their value to that will then introduce further uncertainty. So we can see that it's reasonably consistent for the top three methods, but the bottom method has got a higher uncertainty, probably due to the method that they've used to assign their calibrator value. And what that means is that the overall measurement uncertainty budget is divided into the contributions of those different sources. So the uncertainty of the references, which is this WHO first international standard up here, through the commercial calibrator into the patient sample results. So there's opportunities across the board here to reduce the overall uncertainty, and the onus isn't completely on us within the laboratory. The onus can also be on our IVD manufacturers to improve their calibration, their UCAL, but also on the assigned value of the reference materials as well. So the impact of this is if all of those methods are all using the same reference material, and that has been set at 3%, that is going to propagate through all patient results used across all methods that are metrologically traceable to that standard. And unless a new reference system is introduced with a lower UREF, that's what we have to work with. The upshot of that means is that based on the CRP, the CRP analytical performance specifications, manufacturers have to work to keep the remainder of the variability in the final result down to 2.3%. And they do that in two ways. They do that by minimizing the calibrator uncertainty, as I mentioned earlier, but also by improving imprecision in the laboratory and the imprecision when it's in our hands as final users. And this was nicely shown using sodium and chloride, as I mentioned earlier, as electrolytes that were failing to manage to achieve the analytical performance specifications that were set by changing the reference material or changing the method that the reference material is traceable to. And again, I would recommend this paper by Pascaletti that shows really nicely how the thought processes work with if your uncertainty isn't achieving the standards, how you can go about working with your IVD manufacturers to try and ultimately improve the methodologies that are available to us. And that's where the current understanding of the three models goes. In a nutshell, we have performance specifications which are published, certainly in the chemistry field. We can work out what our local performance is based on that. And if we're struggling to achieve that, we can work out how we can try and minimize that through more than one different mechanism. Analytical performance specifications are now starting to be branched out into different fields. So I just wanted to lead into the final few slides really in the session and discuss some ways that perhaps the Milan models were difficult to apply for very routine activities that we do in the lab. One of which is drug monitoring. So in that previous publication from 2021, I did digoxin was included as the single drug that was being monitored within there. I didn't mention it specifically, but they did suggest that there was a hybrid model that could be used based on the dosing regimens and the elimination half-life of the drug within the body. Because ultimately, if you're thinking about treating a patient with an exogenous pharmacological treatment, biological variation isn't actually what you're measuring. What you're measuring is actually how the biology or the physiology of the patient is causing that level to reduce over time pharmacologically. So it goes back to a model originally produced by by the main man really in biological variation study, Colin Fraser, way back in 1987, combining the elimination half-life of the drug along with the time interval between the doses to permit us to set an analytical performance specification for how drug monitoring assays should perform. And really, that's quite a big step change from what we're thinking about when we're measuring biomarkers and disease processes, trying to diagnose and monitor diseases. This gives us a model to be able to track drug monitoring over a time. Some things have got very narrow therapeutic indexes and have really significant clinical impacts if they're not monitored properly. So therefore, we need to make sure that our assays are working to the standards that we need. And as such, again, hot off the press, very recently in 2023, Cataneo and Pantaghini produced a number of desirable analytical performance specifications based on this hybrid model, using the elimination half-life and the dosing regimens of the drugs. So again, we can see that as time is moving on, we've got more and more information about how the models can be applied, how they can be linked to the clinical impact of the uses of the assays, and it eventually gives us a target that we can set to implement into our internal quality control and our EQA analysis to be able to try and strive to achieve ultimately desirable, but as a minimum, the minimum performance specifications. But ultimately, and all the way through the talk, I've been referring to analytical performance specifications. And the question immediately comes up, especially when we're talking about how the models are applied now and what Model 1 means. Shouldn't we be looking for clinically acceptable analytical performance specifications? And this concept wasn't missed by the group of Rogers et al., a group from Finland and also from Iceland, where they have contributed some further knowledge into combining a lot of the measures that we were talking about before, and I'm trying to quantify diagnostic variation and misclassification. And how that's really changed is they've included pre-analytical variation as well, a notoriously difficult thing to quantify. But they've provided a really nice model where adding biological variation, acceptable analytical variation, and the pre-analytical variation, measuring the impact on the misclassification in different disease states, and from that setting the analytical performance specifications to that whole entire diagnostic process. This is one of the first papers to take that sort of approach. I think this is definitely going to be a direction of travel. I would recommend reading this paper because it really tells us where we are going in the future, particularly with managing decision limits, and because those decision limits, be them reference ranges or single clinical decision limits, ultimately define how we determine misclassification of patients. And now we need to really go back to those initial studies and think, what method and what was the analytical and clinical performance of the methods at the time these decision limits were applied? And I work in hemostasis. In the hemostasis field, I'm doing some work on it just now, where decision limits that are set in stone based on some work that was done 25 years ago might not actually apply as that hard number based on the change in technology now. And so maybe we need to revisit those decision limits based on incorporating the performance specification models as we've discussed today. Of course, we've got to work out from a clinical perspective what even is acceptable clinical performance. Some measurans we're going to want to have a high sensitivity, some will want a high specificity. And these terms, along with many others, are what I'm going to talk about in webinar 3. So please sign up to that. It's coming your way. So with the models that I've mentioned just now and the advances that we've had, obviously, there is still no satisfactory resolution to what one model or what one paradigm we should be using. And this is something that has been in the literature for a long time. And there's an ongoing, maybe battle's too strong a word, but there's an ongoing intellectual discussion about whether the total error or the measurement uncertainty models are the most appropriate, or could they be the one size fits all. I'm not going to choose one side or the other. I don't believe in one over the other particularly. I think what the good thing about all of these discussions is the enhancement of the knowledge and the understanding that we have gained from these discussions has really pushed the science forward. But there's a massive elephant in the room, and I'm just going to finalize the last couple of slides with a little bit of a rant from a hemostasis perspective. Everything that I've presented today and everything that I've shown today has all been, to their absolute credit, based around work done in biochemistry. And there's lots and lots of other disciplines that are trying to play catch up with these analytical performance specifications. I've used serology as an example, mainly because there's been a really interesting series of papers published in CCLM, Clinical Chemistry and Laboratory Medicine, challenging performance specifications in different disciplines based on how the performance of the measurand and what the clinical utility of the measurand is and what we require of it. I think it's really, it's opened up a lot of doors now to us and the other disciplines, where previously, when there was only really one model that we had to pin our hopes on, the total error approach, we knew in hemostasis it just couldn't be applied to us because of the clinical utility of our tests and because of the very poor biological variation data that was published. We're now moving into a situation where we can start thinking about applying performance specifications in different ways and in different activities within the lab as well. So a lot of the discussion around the serology story is massive lot-to-lot variation of internal quality control and how that's managed, and the fact that it can't simply be recalibrated from lot to lot. Because in serology, much like hemostasis, we're looking at biological functional assays rather than inert analytes. So the work that's being done now is really giving us a lot of leverage to be able to think about how to handle this lot-to-lot variation. And that hasn't been missed as well with, again, a publication that's come out very, very recently about taking the uncertainty concept that one step further and subdividing our internal intermediate precision into lot-to-lot variability, and therefore being able to apply an analytical performance specification to what we accept as lot-to-lot variation within our QC. Because if it's too much and we get a shift, we do have a danger of our patient shift results being masked. So I won't go into detail in this in the interest of time, but I will point you towards this publication highlighted on the bottom, which I think is a really, really elegant piece of work that demonstrates how analytical performance specifications aren't just for QC, they're for the entire testing process, and providing this algorithm that we have here, which does contribute biological variation data, we can now have a better handle on which models to use for our tests. Bearing in mind that all models are wrong and it's down to us to decide which ones are best. Often those that shout loudest are not the ones that are actually right. We just have to be able to balance out what is being said in the literature by everybody and actually make our own decisions as to where we go. And that has to be discipline specific as well. But I will say that we are really, really grateful as a biomedical science discipline for the work that has been pushed through in recent years for this. At that point, I'm aware I'm about to hit the 1 hour. I will close. Please, please, please scan your QR code here and give me some feedback, any suggestions, any questions. They will be sent to me. I will be happy to engage. But other than that, all that remains is for me to thank you for your attendance. Please sign up to webinar 3 and I'll hand back to Susan.
Thank you very much, Stephen, and thank you everyone who joined us for that session. Just due to the time, we'll not take any questions live, but as Stephen mentioned, really recommend if you could scan that QR code. There is a section for feedback, for questions, and this will obviously be passed between ourselves and Stephen, and we'll get back to you as soon as we can. Just regarding the session, I hope everyone enjoyed it. We will be This recording will be made available in the next few days, and we will obviously, anyone who was live on this session, we will be sending you a link to rewatch the session. I'm sure everyone enjoyed it and probably take a bit more time with some of those calculations. I know I will myself. And as Stephen has mentioned, we do have one final session coming up, the last webinar of 2023. It will be on the 12th of December, and we are looking at clinical performance acceptables, such as the specificity, sensitivity, as Stephen has mentioned. So again, we'd like to pass the thanks on behalf of everyone back to you, Stephen, and we look forward to hearing from you again on the 12th of December, and hopefully everyone can join us then again. Thank you very much, everybody.
Thanks, everyone.